is a CML case, 15-year-old male. We just know that this patient had marked leukocytosis, and then when we see the differential, we had all kind of this from immature to mature differentiation of the myeloid. So I think yesterday we also talked about some case like this. Our differential maybe kind of reaction due to infection or another thing, or also neoplastic myeloproliferative disorder, including CML. Like the patient we talked about yesterday, the CML. So this is all in our differential. So then for CML diagnosis, the really important thing is we need to know that it's the BCR, ABL, BCR ABL transfusion. So there are also 3 breakpoint cluster for this BCR gene. So we have this big M, that's the major, small m is the minor, and then this micro. So just like we also have this 3 group. So for the first group, big M BCR, the transfusion is related to this exon 13 or 14, and we have this fusion protein 210. We detect this big M BCR in majority of our CML and some B lymphoblastic leukemia or lymphoma patient. And so what I just remember, this is the big M is related more in adult and then CML. And then this small m is related to the exon 1 of the BCR gene, and then had a smaller fusion protein 190. And then it's rare in CML and more related to B-ALL, lymphoblastic leukemia, and then more in pediatric patient. So I remember the small m is more related to pediatric. Of course, in adult patient, we also have 60% of cases. And then for this micro is not that important. They have the largest size of the fusion protein and it's rare. And if we have these BCR ABL, it is associated with the case of neutrophilic CML. So this is a diagram to show how BCR gene and ABL gene just transfusion. So we can see this exon 13, exon 14. So these 2 means that is the major big M BCR. And then we see this exon 1, that is a small m BCR transfusion of this ABL1. Then let's see one of the methods to detect this BCR ABL transfusion. This is a quantitative RT-PCR. So let's read this control first. Every time when we see a test, we need to read a control. So starting from the left, water control, that means you just add water, no any template. So we could not see any band here. If we see any band, the whole test will be destroyed. That means we have some contamination. So and then negative control here. So we can see one band, but not here. Then we have this, I don't know why they use this B3A2, maybe the older naming. So I just label here, this is for the exon 14, this is exon 13 for transfusion of BCR ABL gene. So this is the normal control. We want to see this band. Then if we see this band, that means this control work. Also this one and this one. So, and then we compare these bands to our patients results. Then this is, I think this is a duplication of the same patient. And then we can see the band size are the same of this one. So we know this patient had a big M BCR and then also related to this exon 14. So this is another test, quantitative real-time RT-PCR. So here is a different picture than the previous one, because the previous we usually call the previous one as regular PCR, and then this one is real-time PCR. So we can see this amplification curve. So how to read this kind of picture when you have it in the test? In the test, of course, they won't label it as CD21 here. They will just let you see, I think. So we can see this blue line. So this is just a threshold background infrared. So I think in experiment, you can move this line. But usually the machine will set up like of a background. So we see this background line and then cross with this application curve. So this cross points, and then we go down here, 15, 16, and then until here, we know this is 21. So we know this is CT cycle of 21. And then how about this GUSB? This is internal control. So to tell us whether our template primer or any other thing works, and also a control for how many cDNA we loaded in a sample. So then we know this patient had the BCR able transfusion, and then the CT value is really low. Low CT value means higher expression of your targeted gene. So that means 21 is really low CT value, and then the expression of BCR able is relatively high. Then the patient before our targeted therapy. Then how about here? It seems the whole red line just shift to right, and then we know the CT cycle is higher. Higher CT cycle, that means the expression is lower. But do you think it is low enough if the patient under treatment? It seems not really. During the COVID-19 pandemic, we know we use this real-time PCR to detect the gene expression of the COVID-19 virus, and then usually they set up the negative for the CT value, usually 34, 30, 33, something like this. But this is 29. Definitely, this is really detectable for this gene. So, and then after the treatment for a while, the whole application curve moved to left, CT22. So that means this patient after the treatment, they get resistant. Then they do this single sequencing to find that this is the patient, this is the normal, there's a T, and then mutate to G. So it will cause this for the maybe first generation of TKI resistant, that the CT value is really low, expression really high. Then they used like newer generation of TKI. So they will just make the CT value higher, lower expression of BCR ABL. Then here's a really important table to show you what kind of molecular technique we can use to detect this BCR ABL transfusion. So start from the low sensitivity to the highest sensitivity. Conventional cytogenetic, FISH, quantitative, our regular PCR, we just see the band size running the gel. And then this one is our real-time PCR. So this sensitivity is really important. We know this is
just counting how many zeros.
yes. The reason this is the original table, I just put something like here because this is really useful. We always talk about BCR able, we use log 2, log 3. What does that mean? You just plot how many number after these points. So let's show you what does that mean. So I copy paste this one. 1 log reduction, that means we reduce 90%, and then 2 log, etc., something like this. So we really care about this response after the treatment. So for this one, for us, is what really important for us. For the molecular purpose, we really want to know what is complete cytogenetic response. So that means BCR-ABL after the treatment less than 1%. So compared to this one, this we call the 2 log, 99% reduction. That means less than 1%. And then this is the goal of a TKI therapy. Then we have better response. That is the major molecular response. So that is 3 log. I think remember 3 log may be easier to remember how it's like 0.1%, 1%, or any like this. Log 3, you can just count like this. Reduction of 99.9%. So come back to this one. For this one is 0.001. So there were 3 numbers after this point. So that is 3 log. Remember 3 log, that's the major molecular response. And then we have deep molecular response, that the 4-log, even higher, 4-log that mean like this. Why this is important? Because from the clinician, they can use this deep molecular response to decide whether they can stop the treatment for the patient. So that's
Right.
important.
That's really, really important because previously that's a lifelong treatment. Right? And now you can talk about someone, you can stop the treatment. And even after stop of the treatment, you have to monitor this variable, right? Because it may come back. Yeah.
Yeah.
What's our current detection limit in our lab?
I think I will have use this one.
4.5 log.
So that's really important. So this is just a table I share how tests recommended for monitor response to TKI therapy. I don't want to read this table. You have the PPT, so you can read by yourself if needed. So let's quickly go through some questions. So in this PCR-based detection of PCR ABL gene rearrangement, what kind of product we use? I mean, we extract from the patient and then load it into the machine, what we access. So that is the RNA. What is different with RNA and DNA? That's one thing. The DNA, we have this non-coding region like the in front. RNA, we just everything just coding, so it's shorter. Then what is the statement is incorrect? Let's go one by one to save sometimes. A lower CT value indicates a greater amount of targeted messenger RNA. That is correct. Then endogenous ABL transcription level may be used to normalize. Yes, we can use that. And then CT value of a sample is compared to CT value of various standard in order to determine various standard, including other internal control, that means. And then the final plateau phase of PCR amplification is used to determine the CT value of a sample. We know that that is not correct because we use to see that cross line with our background baseline. Do you remember that blue line? So we use the amplification curve to cross with that baseline. And then we can determine the CT value.